Hello, Life Science. Today we're going to continue Chapter 7, the immune response with Document 2, specific immune response. So please follow along. Let us remember the steps of the immune response. First of all, the natural barriers that are the first line of body defense, when not damaged, they prevent infection. Once one of these barriers is damaged, then there is infection and the non-self enters the body. The immune system here interferes. In a first step, and within few hours, a non-specific immune response is triggered. Then the macrophage tries to eliminate the non-self by phagocytosis, as we have seen in the previous video in Activity 1. Phagocytosis might succeed, but also it might fail. So if the macrophage fails to eliminate the non-self, then here, in a second step, the specific immune response is triggered within several days. And in this case, the effectors of the specific immune response that are going to be activated are the lymphocytes, B and T. So in this document, we're going to explain what is a specific immune response and identify its different types. So the objectives in this document are to show that an immune response against an antigen is specific for this antigen, indicate the two types of specific immune response, and deduce the effector cells or molecules of each type of specific immune response. First of all, let's see why is the immune response called specific. We have already seen in Activity 1 that the first internal line of defense is the non-specific immune response performed by the macrophage, and we have called it non-specific because it doesn't depend on the identity of the intruder. That is, the macrophage is able to eliminate all kinds of intruders, bacteria, viruses, in the same way through phagocytosis. However, in this activity, the second internal line of defense, which is the specific immune response, is completely different. Let's see what do we mean with specific immune response. The experiments performed below in document A, they lead us to understand better the meaning of specific immune response. So, Vibrio cholera and Salmonella are two types of bacteria that cause serious intestinal infections. Then, experimental injections of live Vibrio or live Salmonella to animals are fatal. If they are submitted to high temperatures, these two bacteria, then the bacteria die, yes, but their antigens are preserved. What does that mean? Then, when we expose Vibrio cholera bacteria or Salmonella bacteria to high temperature, then they are killed. Killed Salmonella or killed cholera, these bacteria are now weakened, which means they don't cause harm anymore to the organism, but at the same time, they preserve the antigens they have on their surface. So, which means they are always non-self and they are always stimulating the immune response. So they are considered by the body as non-self, but without causing any harm. And this to see how is the immune response going to be against these antigens. So look at the experiments in document A. We have different hens, experiments 1, 2, 3, 4. And in every experiment, the hen is injected first with a killed antigen, a weakened antigen that doesn't cause harm to this hen, and a few days later with a live antigen. Please, I want you to pause the video for a while. Try to understand these experiments, then we're going to answer these questions. Interpret these experiments, and what can you conclude? So as you know, to do an interpretation, so you should do first an analysis followed by a significance. And to do the analysis, you should indicate the variable factor in the experiment. So if you look at these experiments, you see that the variable factor is the type of antigen or of non-self that is injected, whether it's the same as the weakened one or a different one. So in experiment 1, the hen that is injected with killed salmonella survives when injected 10 days later with live salmonella. However, in the third experiment, when the injection of killed salmonella is followed by another non-self like Vibrio cholera, the hen dies. This shows that killed salmonella protects the hen against live salmonella but not against live Vibrio cholera. Similarly, for experiments 2 and 4, when the hen is previously injected with killed cholera, it survives only after the injection of live Vibrio cholera, while it dies when injected with the live salmonella. So, the injection of killed Vibrio cholera protects the hen only against cholera and not against salmonella. So, to conclude, the immune response triggered against an antigen is specific for this antigen. So, for example, if I receive an injection of a weakened influenza virus, then this means that if at a certain time I catch this virus, then I will be protected against this virus, but not against another virus. So the immune response my body produces is specific for this antigen that I have encountered previously. So the specific immune response, unlike the non-specific immune response, depends on the identity of the intruder. It follows the non-specific immune response. It helps stopping the infection if the non-specific immune response fails. Okay, let's move to the second objective, the types of the specific immune response. So we have two types of specific immune response. We are going to determine these types through these experiments, experiment A and another experiment B. Let's start with experiment A. We study here the immune response against tetanus toxin, that is a toxin secreted by the bacteria Clostridium tetani. And the toxin is responsible for a disease called tetanus, which can be fatal. Laboratory manipulations eliminate the harmful effects of the toxin without altering the antigenic determinants of the bacteria, and the resulting neutralized toxin is called a toxoid. So look at the experiment. You have a mouse A1 injected with the tetanus toxoid. Then later on, blood is taken from this mouse. Serum is separated from cells. The serum is injected to mouse A2. The cells, the blood cells we mean, that like lymphocytes, are injected to mouse A3. Then A2 and A3 both receive the injection of the live tetanus toxin that is fatal. Okay, the A2 mouse survives, the A3 mouse dies. Then the same serum taken from the mouse A1 that is immunized against tetanus is passed through a solid matrix containing the antigen, the tetanus toxoid. And later on, the filtrate is injected to mouse A4, and the mouse A4 dies after an injection of tetanus toxin. Okay, what I have already now done is a description of the experiment, but the question is to interpret this experiment. So again, let's analyze, then draw out the significance. the injection of tetanus toxin into mouse A2 that has received the serum from mouse A1, which is immunized against this toxin, doesn't cause its death, while the injection of toxin into mouse A3 that has received cells from A1 causes its death. Then the variable factor here between A2 and A3 is whether the mouse has received serum or cells. Okay? Then this indicates that the effectors that protect the mouse against tetanus toxin are molecules in the serum and not the cells. So we can see now that the serum has protected the mouse from the tetanus. The injection of tetanus toxin into mouse A4 that has received the filtrate of the serum after being passed through a support containing the antigen tetanus toxoid causes the death of this mouse. This indicates that the effectors of the serum, the effectors in the serum are molecules that can bind to the antigen tetanus toxoid, and actually these molecules, of course, would be the antibodies, anti-tetanus toxin antibodies. Then the serum actually of mouse A1, if we go back to the top of the experiment, the serum of A1 contain antibodies against tetanus toxoid. These antibodies are produced by the mouse A1. Then the mouse A1 fights tetanus toxin through secreting antibodies in the serum against this tetanus toxoid. Okay? So here, this is the first type of specific immune response ensured by the serum. So we have seen in experiment A that when the mouse is injected with tetanus toxin, it fights this toxin by secreting antibodies. You know the antibodies are produced by lymphocyte B. So by secreting antibodies against this toxin. Let's see if the mouse is injected with another type of antigen, what would be its response? Here in experiment B, we study the immune response against the Koch bacillus, which is responsible for tuberculosis. KB causes intracellular infections in the lungs and other organs that might lead to death. C and G Bacillus, CGB, is a harmless bacterium that shares common antigenic determinants with KB. So we can call it weakened KB or killed KB. Okay, look at the experiment and let's do an interpretation. The mouse B2 that has received blood cells of the mouse B1 immunized against KB survives when injected by KB. But mouse B3, which has received the serum from B1, dies when injected by KB. Then, this shows that the effectors of the immune response against Koch bacillus are the blood cells and not molecules in the serum like we saw in experiment A. Mouse B5 that has received T cells from B1 survives when injected with KB. However, mouse B4 that has received B cells dies. This indicates that the effectors of the immune response against KB are the T cells and not the B cells. So in this experiment, when the antigen is a different one, it's KB here, it's not the serum that protected the mouse, it's the cells. And now, can you understand better the meaning of specific? So, now we're done with the interpretation of both experiments. What can you conclude about the type of immune response in each case? Conclusion: the immune response against tetanus toxin, that is in experiment A, is humoral mediated. What do we mean with humoral mediated? This means that it is mediated by humor corresponding to the serum, and thus it is mediated by antibodies released in the plasma. So when the immune response against an antigen is done by antibodies found in the serum, this immune response is called humoral immune response. While the immune response against Koch bacillus that is in experiment B, is cell-mediated immune response, which means it is ensured by cells like T lymphocytes. This is the end of our activity. The main notions in this activity are the specific immune response is of two types. Specific humoral immune response mediated by LB antibodies. Humor means serum or plasma. It is usually directed against extracellular antigens like bacteria. So when the body is infected with an extracellular antigen that doesn't enter your cells like bacteria, so the specific immune response triggered will be mediated by lymphocytes B or by antibodies. The second type of immune response is the specific cellular immune response that is mediated by the cells T8 or Tc, and thus in this case it is usually directed against intracellular antigens like viruses and against grafts. So whenever you are infected with a virus, so the specific immune response triggered will be mediated by the lymphocyte T8 or Tc. The specific immune response develops usually 1 to 2 weeks after contact with the antigen. And don't forget that we called it specific because this type of immune response varies according to the type of antigen. If it's a virus, for example, it is going to be cellular immune response or cell-mediated immune response. And if it's a bacterium, it's going to be humoral immune response or humor-mediated immune response. Thank you for your attention. Stay safe and hope to see you soon.